Okay, so welcome back. So we now switch gears from epidemiology to host immune response. And to speak about the immune system, we have Vineeta Bal, who's is from a medicine background. She's an immunologist who has been with the National Institute of Immunology from 1990. And of course, she has been known to us for a fairly long time. In fact, she was a speaker in the first workshop on modeling infectious diseases we had in 2006. So welcome back, Vineeta.
Okay, thanks, Sitabra. This has been sort of going on, and I have been thinking about what to talk. And before I start, I wanted to ask similar kinds of questions that Sebastian asked, only different discipline. How many of you have done biology for class 12? Ah, great. So I understand that the modern biology for class 12 includes immunology. Is that right? Or you are older than that? I mean, I'm ancient, but I'm sure you are younger. So no notions of immunology and infection were introduced at that point? And do I assume that all of you have a non-biology background thereafter, or some of you have biology background? One. Exception proves the rule. Two. Okay, so it might be even harder task to explain things. However, do feel free to interrupt so that the questions do not accumulate. And between Sitabra and Gautam, responsibility of talking about the biology of infectious diseases has been given to biology and immunology to Satyajit and I. Satyajit is not here today, so he would not know what I'm going to talk about. So while I will address as many questions as I can, tomorrow the sessions of question answers can continue. So that's the understanding that he and I have come to, and I have come here today. So with that, this is a more general title, Invasion of the Host by Microorganisms and Immune Responses. And this essentially involves two components. One is the host, the other is the microorganism or parasite as a general broader category. However, since we are talking about modeling, there are different ways in which this modeling can happen. Oh, this is too tall. Okay. So modeling, and this is not all exhaustive list, but modeling can be to look at the parasite survival versus death. And this is something that people do look at because some parasites survive within the cells within the host immune system or the host, so that which parasites are more susceptible to death, which are less susceptible to death, and why that is the case. Are these factors which are completely and solely dependent on the organism, or is there a role to be played by the host? These factors would come, but the focus would be on the parasite. The second is modeling to look at the individual host. So we are looking at parasites coming to one host and looking at how the host deals with the parasite. So this is host trying to kill the parasite so that the host can survive. This can be human host, and it could also be model host. And I'm using word model with double meaning, and I'll explain that in a while. The third point is, of course, looking at the combination of these effects. And the fourth one is what Sebastian was talking more about, which was modeling population outcomes or population consequences for infectious diseases. So there is morbidity, mortality, and spread and other factors which will have to be taken into account. There can also be modeling for the take of vaccines and or protective immunity. The herd immunity notion was also introduced by him, and that is also useful for determining how vaccines should work, how effective vaccines should be, and also to control the disease. So I will not talk about the first, obviously, but when I talk about host immune responses, these 4 points do get covered to some extent. I'm quite aware that it is not going to be very exhaustive and complete, but let's see how far one can go. So this is what I mean by host-parasite interactions and outcomes. It's very easy to find multiple images of a balance on the web, and this is one of those. I don't remember where, I mean, there was no link that I could easily find. However, there is host defenses to overcome parasite survival, and there is parasite's effort for survival in the host. And I should mention here that some of the parasites can survive outside of a host because there is an intermediate host. As cholera was mentioned, cholera bacteria can survive in saline water or salty water, and they don't necessarily need human host for their survival. However, if you remember in what we do not have as a disease now, which is smallpox, the smallpox virus could only survive for longer durations, if one can call that, in the human host. There was nothing intermediate. So as was being described earlier, if there is one epidemic and followed by a long gap, so that for whatever reason the virus is not reaching the host, then there was a higher chance that the virus would actually simply pop off, I mean, from the world. Not in itself, but it will simply die off. And one reason why the vaccine was so effective against smallpox was this reason, that there outside of the virus and the human host, there was hardly anything that could have helped in survival of the parasite outside the host. So this is an important point, and that is why what the host would like is to just get rid of the parasite, especially the live parasite, and then check how it can survive. And there is a parasite which would want to continue its existence, and that's why it's a balance statement that one has to make and also try to follow. So what I will do first is try to address the host components in this tug of war, and then maybe, I mean, it will get mixed with the parasite, but at the same time, since you have the unfortunate situation of listening to me for next 3-4 hours, tell me if you feel too bored, walk out if you feel bored, but otherwise we'll see how much I can cover as a road to understanding. So really basics, and this is, since I asked about the biology, I'm sure at least this picture you may be familiar with, but this is sort of a broad picture of the immune system. And what it does not cover is blood vessels and the heart, because those of you who work on human infectious diseases are aware that blood is taken as one of the constant samples from the infected individuals, people who are sick, to look at what is happening in the immune system. So blood is obviously in some fashion, not the true fashion, reflection of what is going on in the host. However, this is the immune system spread, and the reason why I like to show this is that you will realize it's all over the body. So what you see here are the bones, and in the bones the T and the B lymphocytes and many, in fact almost all cells of the blood are generated. So these are the hematopoietic systems of the body which are generated in the bone marrow, especially in the postnatal life, which means after birth. There are organs like spleen, this is the thymus, and there are some of the organs which are associated with the gut. So these are called secondary lymphoid organs, and all the small white dots that you see are the lymph nodes. They are given names based on where they are located in the body, but if you can imagine that there is some injury somewhere here on your foot, there are these lines which are drawn which are called lymphatics. Those are the ones which drain the lymphatic cells, which means primarily B cells and T cells, and those are sort of taken away, then they reside, come this far and then go back, and here somewhere in one's neck all this lymphatic drainage goes back to the blood, and then again the blood circulates, and assuming this also might be useful information, that once the heart is pumping the blood out, it goes into the arteries. These arteries end up in the periphery where there are so-called capillaries. From the arteries, the blood, more than the blood, the fluid goes out into what are called the body parts or the tissues, and it is collected by these lymphatics and it's brought back into the circulation. And some of the fluid which doesn't go out of the blood is actually brought back by the veins. So there are arteries, capillaries and lymphatics, and then there are veins coming back to the blood, coming back to the heart. So this is the circulation. So it's a constantly flowing system, and it's also present practically all across the body. And that is in contrast to what you see here, which is a respiratory system, so that we breathe through our nose, then there is windpipe, then the pipe divides and there are lungs where the gaseous exchange takes place, and this is the system. It doesn't really go beyond. So when we define respiratory system, it has relatively speaking localized presence. If you look at the digestive system, again it starts pretty much where the nose is, or the passages go in parallel, and this is the position of the respiratory system, which is not occupied by the digestive system except for this esophagus, or the food which goes down from the mouth up to the stomach, and then there is this complicated system. This in itself is also extremely complicated, but you can see the difference that unlike the immune system, which is really widespread, these two systems are more localized and restricted. The reason why immune system is so widespread is because there is no predicting where the microorganism will come our way. The most common site in which, at which there is exposure is the gut. And we don't think about the gut, but there are our gut, and that is a constant exposure. You should also remember that when a baby is born, so there is mother who has fetus growing inside her body, and when after completion of 9 months, normally full-term pregnancy, the baby is delivered. The baby is as sterile as you can imagine, and when I say sterile, I mean it's practically free of most of the microorganisms that all of us harbor. So that sterile baby, as it gets delivered through the vagina, starts getting small portions or small numbers of bacteria and other organisms, and slowly accumulates into what we know as our excreta. When we pass our motion, it's a solid stool, but a lot of it is actually also consisting of billions of bacteria. So every day there is so much of exposure. And what the immune system does, so this is the host parasite interaction, which is very, very easily seen and can be hard to imagine even for people who work in immune system, but it is such a constant exposure, and I'll also highlight some other areas where these exposures are common, and these exposures actually make it important that the immune system should be ubiquitously present, not just present, but should also be poised in such a manner that it can start activating and responding at any given time point if it is needed. And also it should be remembered that if it unnecessarily starts acting up or responding, it's a waste of effort. So this balance is something that is another one which immune system is trying to straddle. This is not to frighten you, but essentially to list the components of the immune system. This is not an exhausting listing. However, there are sort of two components of the immune system. One is called innate immune system or natural immune system. The other is called as acquired immune system. And you will realize that there are cells and soluble components. Here also there are cells and there are also soluble components like antibodies or immunoglobulins. So I don't want to go through this, but all I am saying is at this point is illustrated in this cartoon. This is sort of a set of organisms, or set of microorganisms, and these are sort of barriers which are there, natural barriers, and there are two broad ways in which the invaders, as I am going to call them, are recognized. So you probably know, some of you might know something called a common organism, common bacterium is called Escherichia coli or E. coli. And E. coli is supposed to be a good bacterium which is normally present in our gut, but it does not cause problems in most human beings. There are also bacteria which have become very popular in the past few years called lactobacilli. These are the bacilli which you can eat through yogurt and other stuff and are supposed to be very good for you. These are the kinds of bacteria which are there, but they are not dangerous. However, there are similar looking bacteria, and when I say looking, somebody had asked about antigenic diversity or something earlier. So all I would say is that there are people, and some of them are more similar, some of them are less similar. So what I'm calling as bacteria which look similar to E. coli, and one category is Salmonella typhi, which causes typhoid in us. There is 75% similarity between E. coli and Salmonella. Salmonella is known to be a pathogen, an organism against which we have to really fight. E. coli one can ignore in most instances. So what is shown here is that when these organisms are there, the early immune response is mounted through what are called as clonally uniform defenders, as against late responses which are called as clonally diverse defenders. And I'll name them, not necessarily that you have to remember, but if you're going to model infectious diseases, you can avoid looking at the specifics as long as you know what compromises you're going to make. And in that sense, what Sebastian showed as Oxford Dictionary definition or something of modeling was important, that it simplifies the matter. There are certain things that have to be taken cognizance of and certain things which can be ignored. What mathematicians would like to ignore and what an immunologist would like to ignore are debatable issues. However, models are models. So you take certain things for granted and you ignore other things, and that's why they are models. They are never perfect scenario, and if you are going to model, if you are going to look at human infectious diseases, remember that modeling is an approximation. It will have its own values, but it's an approximation. So here the first set of cells which will respond first are the ones which will sort of will recognize the red flags, so to say, on different organisms, because those are the danger flags. So the other organisms which are not flagged will not be recognized as danger. However, certain others will be recognized as dangers, and all the dangers look pretty similar to each other. Not identical, but they have some similarity. You might have heard something called toll-like receptors, TLRs, which are flagged by many such organisms, and TLR is a big family of receptors, and such receptors are one of the flags which can be easily picked up by the cells of the immune system to start mounting an immune response. And I'll take you through this, why these responses are equal as important as what are the clonally diverse defenders. So clonally diverse defenders are comprising of what we call as adaptive immune cells, T cells and B cells. And the reason why there are different colors there is that if I had a possibility of showing 10 million colors, that's the number of T lymphocytes you have. And each color represents ability to recognize one such shape or one such organism or one antigen, as immunologists would call it. So in adult human beings, there are really 10 million different T cells and 10 million, approximately the same numbers, 10 million different B cells which are there. So you can imagine that somebody not recognizing an invader will be an exception rather than the rule. If there are such a diverse populations available, then chance that something is recognizing something is more or less statistically, you should know, statistically it's unlikely that any organism would go unnoticed. So if we are looking at that frequency, you also will realize that if I have to match a blue color organism with a blue cell out of 10 million different cells of different colors, then it's going to take time. So that early window when this matching has not happened, this is the first ever exposure that I am talking about, what we call as primary immune response. So babies, for example, when they are exposed to measles virus or something else, those are likely to be the first exposures, especially in the childhood. So if this blue organism has to find its target, or actually this blue cell has to find its target and start responding so that the blue colored organism does not go haywire and establishes itself, then you can imagine that finding this pairing will take time. That time is actually the most risky time, and that risky time is covered by these clonally uniform defenders, what we call components of the natural immune system or innate immune system. They will provide protection for the first few days after the exposure. And after about 7 days or so, this matchmaking has happened and the cells have proliferated and are present in large enough numbers now, so that there is possibility of immune memory, which is something that we are talking, Sebastian was referring to in terms of protection, in terms of vaccination and many other aspects which are extremely relevant in the in combating the infection. So this is, as I said, are the two components of the immune system which come into action and are dependent on each other. So for example, if these red flagged cells are taken up by cells of the phagocytes, which are the cells which pick up particulate matter, and these cells are called neutrophils or monocyte macrophages, then these cells would phagocytose and kill the bacteria, and we'll look at it as an immune strategy at a later time point. And these cells, after killing the bacteria, would have proteolytic enzymes acting on the organisms so that they are chewed up. So essentially, small peptide stretches from the whole organism, or lipopeptides or carbohydrates, will be generated in the process of phagocytosis followed by enzymatic degradation. So that's the process that is taken up or done very effectively by such cells, and such bacterial products which are residing in these cells are the ones which are eventually recognized by the T cells as well as B cells, and we'll come to that in a while. So these are the two components, both are essential, and human beings which are defective in either one or the other are invariably compromised and face recurrent infections, and mortality or death is quite high in such immunodeficient individuals. This is another way of showing, and sorry for keeping quoting this series, but Satyajit and I in historical times, really 20 years ago, had written articles in Resonance, and Resonance was a new journal at that time relatively, and we had written 5 or 6 articles essentially keeping class 12 students as the target audience to introduce immune system to the audience, to the readers, and sort of hoping that people will get interested in the immunology research. However, those are available with these 2 guys as a PDF, not necessarily in the best shape, but they are available, and if you feel, you can look at it, because some of the logic, in fact large chunks of the logic that I am going to explain to you today is primarily based there, which is modified further. So this was the way we wanted to predict immune system function, that it has to be everywhere. And earlier I said that gut, which is not necessarily shown here, has large number of bacteria and other organisms, also viruses also present in the gut. But in addition to that, the organisms are everywhere. Some people are very finicky and they go through cleaning their fingers and everything repeatedly before eating food. So that's a good habit, so that the load decreases. But the television ads that you see, 99.99%, you will know much better than I do that if you have only 1,000 bacteria on your skin, that 99.99% may leave a few bacteria. But if you have billions of bacteria, then that 99.99 would still leave thousands and thousands of bacteria. So while the claim may not be wrong completely, it is meaningless. So that doesn't mean that you go and dip your hands in the soil and start eating, but people like me, yes, I know, but people like me are immunologists, practicing immunologists, and I still like to eat street food and I survive. That's because I have been growing like this. I was not brought up on what is called distillery water, so that drinking from the tap is something that I still routinely do, and as a result there is this low dose exposure that keeps coming my way and develops gives me reasonable protection against large number of gut microbiome, as it is called, as well as gut pathogens, apart from other things. So I'm not recommending you to do that immediately, but I know there is a next generation, not just mine, but maybe even yours, which is brought up on this clean water business. And if anybody is interested, read something called Hygiene Hypothesis, which will tell you the problems which are associated with drinking distillery water. People who come to us from the foreign countries are advised not to eat salads here, not to drink this water. That is because they are really too susceptible. And there are advantages to them and advantages to us. We shouldn't be losing our advantages and simply getting disadvantages. That was a side issue. However, all I wanted to point out was that the bacteria are present inside the nose, inside the mouth, and anywhere you care to think of. So you can't get rid of them, and that is why it is better to have an appropriate strategy to keep them in certain locations and not come down with the disease. And I'm going to show you some cartoons simply to highlight how complicated immune system is, and it's only to sort of scare you, nothing more than that. That you don't have to get into this, but people like us, or Jose, who doesn't do any bench work any longer or wet lab any longer, but when he did that, he was quite aware and was dabbling into things of this kind. Satyajit and I still do, but this is one cartoon which is about the gut lumen. So this is the gut lumen, this is the lining of the gut, and this is one of the lymph nodes below the gut. Simplified notions, but still. So what happens is there are tons and tons of bacteria tons in terms of numbers, okay, not in terms of weight, but there are really large number of bacteria which are floating all over, and some cells here and there would be capable of sensing their presence. However, E. coli in normal conditions will not traverse this barrier, and Salmonella typhi, despite 75% similarity with E. coli, would easily invade. So, there are some bacteria which will not invade and some bacteria which will invade, and once they invade, they can easily go through all of this circuit, can go through the veins and wander around and land in the spleen or in the other lymph nodes and so on and so forth. So, once this gut barrier is breached or invaded, then it is very easy for such bacteria to reach all sorts of places. And that is where the importance of the connectivity is, but it can be a complete risk. And slightly larger version of the gut barrier is here, that this was the lymph node that I was sort of talking about, and these are the villi. And normally, despite having millions and millions of bacteria there, most of them do not invade, because what we have is what are called sort of barrier functions which are operative, in the sense there are organisms here, but there is also mucus layer. When we have runny nose, we experience mucus layer from another organ coming down, but there is a permanent mucus layer which is present and lining all these so-called exterior of the body, in the sense gut lumen, and because of that most bacteria do not manage to reach the surface of the gut. This lining has a thick coat of mucus, and that helps in preventing the invasion. However, whatever invades, ultimately that they have these organisms have ways of cutting across the mucus barrier and then going inside, but once inside there are many events which will come up. So, this is about the gut barrier. We were talking earlier about malaria in the context of modeling and insect bites. So, we are talking about mosquito bite here. So, this is the cross section, not cross section, this is a model of the skin, because the vessels are never like this. These can only be drawn in this fashion, but this is the outermost layer of the skin, this is the inner layer of the skin, and what is described as dermis from this point to here is the one where you will find vasculature and lymphoids, draining systems very abundantly present. So, a mosquito bite can only go through the epidermis and deposit, say, malarial parasite in the topmost layer of the skin. That is less risky. However, it has also been shown that mosquito bites can also go deep enough to have a direct insertion into the vasculature, into the blood vessels. If that happens, you can imagine the incubation period varying. So, same number of, say, 10⁵ Japanese encephalitis virus or malarial parasite or something or the other, a arbitrary number. So, those either get deposited there or get deposited here, and from that infectious bite the person can come down at different time points with illness. And that will also depend not just on the dose of the microorganism or infectious agent which is introduced, and this is something that was not really discussed in terms of modeling, but it is one of the variables that also needs to be included, and whether such a dose is going into the subcutaneous space below the skin, so that it will take that much time for the organisms to reach the blood vessels and then get distributed, or if it gets introduced directly, it is just not just the speed, but you can also imagine all of those JE virus particles, for example, or dengue particles or some other particles are directly into the blood, and they can distribute very, very, very rapidly throughout the body. So the kinetics of incubation period followed by the infection, actual infection, where there are symptoms most of the times, that period also varies. So when you are thinking of modeling, these are issues which need to be taken into account. There is no uniformity in which the vector will introduce the organism. Numbers will vary, location would vary, how they are reaching, wherever they are supposed to reach, that time also will vary. So this is, I can possibly skip the next one. This was another cartoon simply to talk about the liver, because that's also getting targeted. Oh, okay. The reason why I suddenly remembered. The reason why I had picked up this cartoon was it says Salmonella typhimurium. Salmonella typhimurium is a cousin of Salmonella typhi. Salmonella typhi causes typhoid in human beings very, very efficiently. Typhimurium causes only gastrointestinal problem, as in diarrhea, dysentery. However, typhimurium causes typhoid-like disease in the model system, which I use very frequently, which is the mouse model. So if I am talking about mouse typhoid, I should be using Salmonella typhimurium. If anybody is going to model mouse typhoid, it should be mice as the host and typhimurium as the organism, and one can still learn lessons from this to be extrapolated into human host and Salmonella typhi with caveats. But that's what I mean, that from models one can actually get data which can be used to model human diseases. So there are advantages of using different sets of models as well. So this is again the gut lumen, and this is the lining, and normally very small molecular weight substances can go through the barrier, because it's a barrier which is continuous. However, organisms like Salmonella typhimurium can actually find their way through the gut barrier, and that doesn't mean the barrier is completely damaged. It doesn't mean that the rest of the bacteria which are there in the gut would find a good channel and all of them would get in. It is not. It is very, in a sense, very specific invasion that will come in, but once that happens, there can be for the bacteria to reach liver, to reach spleen and other lymphoid organs is not a difficult problem. So this, as I said, I was trying to give you a sense of how immune system seems to work. This is another leg of that original figure that I had shown you, but to tell you that if there is a site of invasion here, we normally, there is injury to the foot, and of course the dust goes in if you are wearing sandals or chappals rather than the shoes, and then sometimes there is infection. It may look like it's a minor infection, but that's also host parasite interaction and immune response generation. And what people will find is that there is hardly anything on that foot that you can notice a day later, but if there is some infection which is set up, you might have pain in the groin. So the lymph nodes in the groin in this area become swollen. So there was no injury here, there was injury right there. So how did things move? And that's how the immune system starts responding. So there is an invasion or breach in the barrier in the skin somewhere here or wherever, and from there via lymphatic the bacteria or the infectious agents will travel, or if by some chance and good fortune, if there are phagocytic cells which are present there, so that neutrophils or monocytes would actually simply engulf what has come in, and then these cells would slowly migrate through these lymphatic channels to the first set of lymph nodes, draining lymph nodes, followed by another set of draining lymph nodes, or wherever the injury is, appropriately the draining lymph nodes will be there. I am sure you also have noticed that if you are coughing, sometimes you also have pain here, and those are also the swollen lymph nodes which will disappear as the infection or the cough subsides. So, all of these are signs of inflammation and infection which we normally do not realize, but that is the way immune system starts mounting the immune responses. So, these are, as I said, various ways in which the immune system will start responding. And now let me look at what are the outcomes. I gave you a response. Okay, so neutrophils, I am sure all of you at some point of time have given blood. If nobody has ever given blood because you were unwell, damn lucky. I hope there are many more of you like that. But if you give blood and if you get what is reported as TLC, DLC, total leukocyte count, total white cell count, in that you will get a number. It would be between 5,000 to 10,000-15,000 per cubic millimeter. That is the total white blood cell count in your blood, and that is the normal range. And you also will appreciate that this normal range is the mean plus minus 2 SD normally of the population. So, it is a normally distributed population and the ranges are normally around that. So, if you have 2000 as the white blood cell count, then people might suspect that you have something problematic. If you have a count of say 20,000, people will also worry that maybe you have infection and or some other problems and you need to be investigated further. So, this is the total white cell count. There is also some other heading called differential count, DLC. So, differential leukocyte count gives an idea about how many cells there are. There is neutrophils or polymorphonuclear cells, that is one category. This is a granular cell, and majority of the, well, significant proportion, 40 to 60% of the human blood has neutrophil count, as in, I am sorry, backtrack. Out of the total white cells, 40 to 60% are neutrophils. Then there are lymphocytes which can vary as 20-30%. Then there are monocyte macrophages, and then there are small proportion of other cells. So, lymphocytes in this category include T and B lymphocytes. They are not distinguishable just by looking under the microscope, and this is the light microscope version that I am talking about. Just stain, a blood smear is done, stained, and you look down the microscope, and based on the morphology these cells are identified. So, that is how the cell identification is done, but both neutrophils and monocytes are the major cells which pick up particulate matter. So, if there is a danger which is a solution, for example, a chemical solution which is injected into the body for whatever reason, these cells are unlikely to detect it. But if it is a particulate matter, even if it is an innocuous particulate matter, it will be detected because these cells pick up particles. So, these particles will be picked up, and that is why they are called phagocytes. They eat up what they see in front of them, and the neutrophils are the first line of defense. They are really large in number, in significant proportion, and they move in and out of the blood vessel very easily. So, based on some chemical signals that are emanating from the site where the injury or the attack is, the neutrophils would move to that location very fast. So, sometimes within 3 minutes you see neutrophils coming out and getting accumulated. In medical college, I remember there was simple experiment that we were asked to do, horrid ones, but take a safety pin and scratch each other's arm and notice what happens to the injury. So, what you see is, of course, there is a red line because you have gone through the skin. After that, there is a swelling. Sometimes there is a pain also, depending on how deep the cut is. But all of these, the swelling indicates that the fluid has accumulated. Slowly that fluid will also attract further cellular components to it. So when you see swelling at the site like that, that's the cellular component, so that the cells are already there within no time to engulf and clear the zone, if you want to call it that. So first line of defense, cellular defense, is the neutrophils. Once neutrophils are there, they have a half-life of 24 hours to 48 hours, so they will come and die at the site. Followed by that is the monocyte macrophages. So they will also come and do the remaining cleanup. So the phagocytic lineages are monocytes, neutrophils and monocytes in the order of appearance. Monocytes are also called macrophages in different ways. So these two cells would be there, and those are recorded as N as in neutrophils and M as monocyte macrophages in your differential count. So we do have a sense of what these cells are in the count, and those are the ones which help in different ways. So lymphocytes, as I said, are there obviously, and their function will be apparent as we go along. So I haven't talked about this. I will. I was distracted because Gautam asked a question, but that is the reason why I put it up. Okay, so let's talk about different outcomes when an invader. Now I'm not talking about non-dangerous organisms, I'm talking about danger, which means invasion. Some kind of breach in the barrier and the red flags or whichever flags that I had shown you earlier. So the immune system needs to respond. And at that point, the organisms are generally clubbed as pathogen because they cause pathology. So pathogens will be there, and if there is no invasion, which is what I described just now, we are not going to talk about it further because it's possible. Sometimes I may have Salmonella typhi in my gut and it simply does not invade for a variety of reasons, including their numbers may be small. I may have phenomenally large immune response in the gut against Salmonella typhi, so there is no invasion, no consequences, let's not talk about it. But if there is invasion of one kind of barrier or another, then we need to think about the consequences. So first, as I said, there is invasion, and invasion will be associated with neutrophil and macrophages going there, which means there is initiation of immune response. Okay? Because this will be followed by T cells, B cells, and so on. We will try and see if I can cover that. But invasion would mean infection. And here, modeling infectious diseases, when we talk about, there are two things which are there in the title. There's infection and disease, and those two are distinct, and I will actually make that point on this slide. So there is infection, but there is no disease. It may look odd because many people who are not necessarily biologists or not necessarily trained as physicians realize this. Most of us actually do not come down with disease despite infection. You might have heard that there is Japanese encephalitis epidemic somewhere or the other, and how many other people are actually infected and not complained. Those are falling into this category. Infection, no disease. When we complain that I have body ache, maybe I have this pain here, that pain here, that's the disease. Okay? So infection is not disease. In the infectious disease context, infection is much more rampant and what we call as subclinical infection. Clinical means we feel like going and seeing a doctor. We don't necessarily do it, but if we feel that we are unwell enough that we need some treatment, need something to be done, investigations and or treatment, that's the disease. And if, for example, I can quote numbers for Japanese encephalitis infection or Japanese encephalitis virus, there are 250 cases at least before one patient is one patient comes and complains. So the ratio of subclinical infection to disease is 250:1. And for different diseases, for different infections, this is really, really different. And what unfortunately in a country like India we do not know is what are these ratios. There are some ad hoc estimates that we live by, but we don't really know. Even this 250:1 number that I am telling you is not necessarily accurate, because we do not have a reporting system of infectious diseases, so that everybody who is feeling a little unwell or that there is blood is taken and investigations done even from putatively healthy people, so that we know that they were earlier infected with dengue or Japanese encephalitis or polio or something or the other. So in a country like India, unlike the data that Sebastian repeatedly showed about England and measles, they have a phenomenally great reporting system. Many developed countries have, and we don't have. So we have to live in this approximation of what is infection and what is disease. But as a result, most infections come our way and go. We mount an immune response, we have a pre-existing response without realizing that there is a second encounter and a third encounter and a fourth encounter. So this is something that's an important point to remember. So whether it is invasion, infection, which results in no disease, or whether it results in disease, the immune response is generated regardless. And the T cell responses and B cell responses will be there. So without us realizing, we are quote unquote immune to many diseases as we grow older. And contrary to this, if you are, forgive me for calling the Bisleri generation, but if you are belonging to the Bisleri generation, then you haven't had these many exposures as much, especially the gut related exposures, and that is why you will have fewer antibodies or T cell responses which are already there in you, so that you will be more susceptible to infections when it really hits you. Okay? So that's the difference between infection and disease, which needs to be noted and has to be remembered even when you are modeling, because infection is not what you want to model. You want to think, based on the earlier talk, what are the consequences of the disease. But for that you need to know how many people are infected. So when there is that susceptible population, infected population, recovered population, in that infection and disease are not separated, and sometimes infections and disease need to be separated, because otherwise you don't know who is recovered and who is likely to be susceptible in the next round as the models get complicated. So from the disease there could be different outcomes again. So from disease there could be complete recovery, which is very, 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 very common, and that's what we call as not from disease. That is also complete recovery in many cases. For example, flu. Even when there is body ache and something or the other happens, only when there was SARS scare, people talk of influenza or some flu virus related mortality. But in young adults, it's not very common to see disease, death especially, or any of these outcomes from, say, flu. But if you look at tuberculosis, about which you are going to hear later, and if I reach that stage today, maybe we will also talk about it. But if you are talking about disease, then complete recovery versus chronic infection versus carrier stage and death, proportions would be different, and depending on the disease. So there isn't any generalization that one can talk about. But tuberculosis, in a sense, especially because it goes down, it comes back again, it can be falling into the category of chronic disease. It could also be falling into category of what is called as recurrent disease. Depends on what phrases you use, and sometimes something is more in fashion over a period, sometimes something else is in fashion. So when we talk about chronic infection, many of you probably know that if there is chronic infection due to hepatitis B virus, for example, it is very seriously associated with hepatocellular carcinoma. So chronic infection with an infectious agent can lead to repeated attacks on the body, in a sense weakening of the immune system for reasons just now I am not going to get into, but that may lead to the cancerous development. These are some of the possibilities, but as I said, it is not so common to find every infection leading to chronic infection, and many of these phases, chronic infection for example, may not be remaining at a disease state. Okay. Tuberculosis for example, when people talk of recurrence, the person has had BCG or exposure and a lung infection for TB, treated, recovered, and at some point it comes up. Does it come up again because there was no immunity, because there was reinfection or something else, or was there a low level chronic infection there all the time without making the person feel sick? Okay. All sorts of possibilities are there. I am hand waving it only to tell you that you need to figure out about the disease you are interested in and read every detail of this kind. It is not possible for me to go through all of these, nor do I know about every disease, especially the numbers, have no idea. And death is one of the known outcomes for many diseases. Carrier state is an odd entity, uncommon, but hepatitis B surface antigen again is hepatitis B virus can remain as a carrier, host can carry hepatitis B virus and still live relatively peacefully. There was also, those of you who are interested in reading or have read some of the science history would know about typhoid Mary, and that typhoid Mary was a nurse called Mary who had Salmonella typhi carrier state, and she infected many others with typhoid. Yeah. It can be, but I actually avoided drawing multiple arrows. So it is possible that you will have a maybe, may not be. The disease stage is an acute stage or actually illness stage, and you may have recovered from that, but so that the clinical illness has gone away, you may still harbor the infection, and that can be chronic infection. That is one way of looking at it. The other could be, just one sec, the other could be that from infection which is an acute exposure and then you are developing immune response, the immune response is so poor, for example, that you never cleared the infection, but it was not so bad, the infection was not so bad that you never actually developed a disease. So both are possible. So I could have drawn an arrow from infection to chronic infection as well as the arrow that I have drawn. Both are possible. Answered your question too?
Okay.
Yeah. What is there? Okay. So I have put here prevention and or protection as just a notion, and that is about vaccination in one sense, but also protection and immune responses are different. And this is something that I might as well elaborate here and possibly stop before I go to the next one, since I looked at the clock and it is close to 1 o'clock. So what is there is that, as I said, from infection, or if I go back one, no, sorry. Even if there is invasion and infection, there will be an immune response, right? That does not mean that the immune response that you have is necessarily protective, in the sense that next time that particular organism, let's take Salmonella typhi as an example, that I have encountered Salmonella typhi, I have infection and I have mounted an immune response. It hasn't resulted in disease, fortunately, but there was an infection and immune response is generated. This is what we call the clonally diverse, the colored cells that I had talked about, T cells and B cells. Those are the cells which will generate immune memory, and this immune memory is supposed to provide us protection from the next assault by Salmonella typhi. Okay, that's the peculiarity of the adaptive immune system, why it is considered so important, that once you have an encounter, you will be able to handle the next encounter better. So that's the strength of the immune memory. So infection can actually provide you with an immune memory such that even if you have 10-fold higher Salmonella typhi dose as compared to first response, you may not come down with disease. That doesn't mean you will not be infected again. There might be another breach, there might be another trigger of immune response, you will still reach this stage, infection, and may not lead to disease. First time, maybe 100 Salmonella typhi you had and there was no disease. If you had 1,000, maybe it would have gone to disease. It also is a dose, how easily and effectively the immune system can control and contain the infection. If next time you may have 1,000 Salmonella typhi and still not reach this stage because you have immune memory. Okay? That's the advantage of having immune memory, but at the same time, it's possible that whatever has been triggered as an immune response may not be protective. What do I mean by protective immune response? And I'll just now take example of malaria. Malaria is endemic in India in certain big patches, not everywhere, and if it becomes endemic all over the country, I won't be surprised. But the common antibody response that is detectable against malaria is against circumsporozoite protein, CSP protein. Okay? This antibody response is rampant, it's not protective. If you ask me, I don't really have an answer to give you what kind of immune response against malaria is protective. But this antibody response against one protein of malaria, which is an easily accessible protein, so you can imagine that if a protein is present in the shell of an organism, it will be very easily accessible to the immune system. Quickly antibody response is generated, and that's how anti-CSP antibody response is very easily generated in most hosts. In endemic areas, you will find CSP antibody presence in many very high frequency individuals. However, that is not protective. So, for example, if I purify anti-CSP antibodies from individual who has antibodies and transfer it to somebody who is freshly entering the endemic area, so obviously is very vulnerable to come down with malaria, nothing will happen. The person will get malaria if he or she gets enough exposure. So, having immune response and having protective immune response are two categories. They overlap, but they don't necessarily get equated. You may have 15 different proteins of a given organism against which antibodies and T cells are generated in the host, and you may need only one or two of those which will provide protection. Some individuals may mount very strong response against those one or two, and hence will be protected from subsequent infection and disease, but the other 12 or 13 in a sense are sort of also ran. What does that tell you? That immune system doesn't distinguish between what is a protective protein, response versus a non-protective protein response or lipid response or whichever response. It simply recognizes it as part and parcel of the danger. What will be killing or what would be useful for clearing the infection is something that's very different. Two get equated very often, and that is why when people are thinking about vaccines, they are simply reading out immune responses. What is critical to check whether those are protective immune responses is a different ball game altogether. So unless we know what, for example, I won't take malaria as an example because I don't know what is important in malaria, but say antibody against, there are neutralizing antibodies against certain viruses. So detecting neutralizing antibody titer is a good indicator of protective immune response. Detecting simply antibody titer is not. That's the point that I wanted to make, and that point is extremely relevant when people are thinking of either prevention by vaccines and the use of vaccines so that there will be protection against the disease. And mind you, I am not calling it protection against infection, I am calling it protection against disease. Probably I will stop there and will continue. But I am happy to answer questions if you people are not very hungry. It's not so easy to give one line answer for tuberculosis. So if I find time, since I have been given an additional class, we will probably reach tuberculosis. So if I reach there, I will try and explain situation because it is really, really complicated. Gwen is going to talk about the modeling again, but I will, it's as bad differently as say malarial disease or filarial disease, but we will have to handle it more in depth. If we don't do it, then I can talk to you tomorrow. Question is, how do I know every? For example, there are thousands of kinds of bacteria in the gut. At some point, they might have invaded. But do I have a list of 1000 organisms and am I going to check my sera against 1000 organisms? Theoretically, it's doable. Practically, it is not doable. But in clinical situations, when people are looking at seroepidemiology as it is called, there are these chip-based assays which are done now. So sometimes either at mRNA level or sometimes at in some other fashion, the antibodies or immune responses against say 50 viruses are detectable or rather screened for. So if we have wherewithal, which includes a lot of money, then this can be done. And question will again come up that say if I was exposed many decades ago to an infection and was never exposed to that afterwards, would I still show the mark of that infection in my antibody response or in my T cell response? These questions are theoretically addressable, practically not done. So in principle, yes. Many antibody specificities will be detectable in my blood, probably more so than your blood, and certainly a lot more than a 1-year-old baby. And these will be there, so also they're in the T cells, but how long these antibodies will last is another question. And that's not just the protein molecule as an antibody, but the cells which produce those, which are called plasma cells, how long a plasma cell which was generated in my body when I was 1 year old would last so that that particular antibody will be detectable today is a question we have not managed to answer satisfactorily as immunology. Sorry, we may lose, we do lose. And if I talk about tuberculosis, I will talk about it.
Maybe we should stop now because we have a shortish lunch break. Vineeta will be back for round 2 at 2 o'clock. And a few of you have not registered because you came a little late. There'll be a registration counter open at 2 just before we start again. So you can just come back and check at that time. So let's go for lunch. Follow the student coordinators or whoever it was who volunteers in the direction of lunch. There are 3 or 4 of them there. It's just across the road.